Oh, you're itching from your back, yeah.
It had gone away. You know?
We feel bad. Now you have to do it again. Really? That's what we did. Yeah. All right. After many years, our last chapter. Chapter 10, the immoral immune response. We're definitely on the, kind of the downhill part of the book. Most of the tedious stuff is done, luckily. As you can probably tell, this chapter is pretty straightforward, and, there's not a ton to memorize, just some basic concepts to kind of get down. At this point, you should probably have most of what you need to memorize for the boards. Hopefully, if you kind of kept notes or kept track of some of these tables of things that you needed to cram in, most of that should be complete at this point. Now it's just kind of getting the big picture. I think the next chapter is actually about the mucosal immune system, of how all these things kind of go together, but I've tried to, go over that system over and over and over, so hopefully most of the concept is not new to you at this point. Let's see, dynamics of the adaptive immune response. Yeah, again, just kind of big, broad concepts. And there's only, I think, 6 more chapters. So Nikki will go over. The immune deficiency stuff should be pretty second nature, of course, just to review for you at this point. I think there's a chapter about allergic disease, which will, again, put it all together, but most of it will probably be fairly intuitive to you at this point. Autoimmune, actually, I'll take it back. There is, on the board traditionally, there's been a ton of autoimmune stuff, although I think I heard rumor that they're taking it off the boards. Ask Dr. Portnoy about that. So maybe one more list to memorize is all the autoantibodies that go, unless it's officially off the boards, clarify that before you spend the time to memorize those. Although, I mean, they're good to know clinically, of course. But there will be some genes. If you need to know the autoimmune stuff, there's some genes you need to know, right? That would predisposition you to these diseases, and then there's all the autoantibodies that we know. I don't think you need to know too much about the tumor defense. Yeah, you guys are really close to just being done. So, congrats.
I don't remember.
No one in history world has ever remembered this the first time through. You don't think you do. But then when you go through the second time, don't you think when you read it the second time, though, you kind of picked up more than you thought you did the first time through? Yeah. So then once the big picture solidified, then right towards the end of your second year, it's just knowing those tables to memorize, cramming it all in, and that's it. But if you feel completely like you did not learn a thing this entire year, that is, everyone feels that way after the first year. And then once you get in the second year, you realize, oh, okay, well, I kind of get this now, and it's all coming together, and don't worry about it. You are good. Okay. Right, so the humoral immune response. So when you talk about the humoral immune response, we're differentiating this from the cellular response, right? The cellular response is the T-cell responses, and the humoral response is the antibody response. So the humors, right, were this unknown substance that could be transferred between people that conferred immunity. Right now we know those humors are antibodies. So both of those two things together are the adaptive immune response, as opposed to the innate immune response, right? So this is adaptive, and it's the humoral wing, right? And it has to do with antibodies. So antibodies primarily have 3 jobs. So they neutralize, they opsonize, and they activate complement. Okay, so this all gets started in the secondary lymphoid organs, where the B cells will interact with the T cell that activates them, and this essentially has to happen, although we'll talk about a couple exceptions. And then the B cell can actually turn into a plasma-secreting cell, secreting the antibodies, which then can neutralize by binding to an antigen and preventing it from doing whatever it does to infect and harm the body. It can help with opsonization, in other words, that it will bind to the antibody, and then the Fc portion of the antibody can bind to separate receptors on phagocytes and help it know to engulf. And then, of course, a very important part is that it activates complement, which we've talked about before, in that it will bind to the antigen, and this Fc portion of the antibody will start the complement cascade, which, of course, punches holes and kills, or in its own right can opsonize. It can promote phagocytosis. Okay, so antibodies do have two special things that happen that's different than T cells. Once they get activated, they can undergo something called affinity maturation. So that variable region of the antibody can actually become more specific for the antigen that it's fighting. And this happens by a series of somatic hypermutations. So it actually goes back to the DNA, so where that rearrangement occurred, and through the aid of AID, through the help of something called AID, and we touched on this a little bit before, it allows some nucleotide insertions to change that variable region, some of which become more specific and some of which become less specific. The less specific ones, of course, die by apoptosis. And then antibodies uniquely class switch. So the DNA is again activated and rearranged again. Instead of making IgM, it can change that tail, and it can change it to, of course, IgG, IgA, or IgE. So the vast majority and the best form of antibody is made in a thymic-dependent way. So this has to happen by a B cell first recognizing an antigen, and of course it recognizes antigen on its B cell receptor, which is essentially an immunoglobulin that's attached to the surface. It engulfs, it puts it on an MHC, and of course it displays on the MHC so it can communicate with a T cell, like all things communicate with T cell. Essential to this interaction, so you not only have your T cell receptor interacting with MHC and then the co-receptor, which we've talked about many times. An essential interaction is CD40 on the B cell interacting with CD40 ligand on the T cell. I have yet to come up with a great mnemonic of how you remember which is on which cell, and I always forget. So if someone comes up with a good one, let me know immediately. And then there are a couple circumstances where you can actually generate some antibody in a thymic, secreted antibody in a thymic independent manner through cross-linking or through engagement of these pattern recognition receptors on the outside of the cell. We know this can happen because you can see in someone who doesn't have a thymus, like complete George, you actually see some antibody, mostly IgM, not a lot, and not enough to sustain life. So, right, children would typically succumb by 18 months if this is all they had for their defense. Okay. So again, this system is very, very important in that the B cell, when it's in the lymph node, first has to see the antigen. So if you recall, the T cell has already been activated over in the T cell zone. So it's seen the antigen presented on a dendritic cell. But at the same time, that antigen is coming through the lymph, just as the dendritic cell is coming through the lymph, and it gets filtered through the lymph node, and it gets stuck on something called follicular dendritic cells, which are not related to the dendritic cells that are going to display to T cells. But these little bits of antigen will get recognized on the B cell receptor, and it gets engulfed, and it has to get processed and presented. And remember that whole processing pathway we talked about, held up on an MHC, where then a T cell is going to have this interaction with the T cell receptor, B cell receptor, CD40 ligand, and then the T cell is going to be critical in secreting the cytokines to tell the B cell what to do at this point. So the dendritic cell has given certain signals to the T cell, telling the T cell kind of the nature of the infection. And the T cell, in turn, is going to tell the B cell the nature of the infection by the cytokines it's going to secrete. So this is going to cause the B cell to start proliferating, and then it's got a choice of either becoming a long-lived memory cell or becoming a plasma cell. So we know these memory cells, or I'm sorry, a memory cell, or also a long-lived plasma cell. And we know these plasma cells can stick around a long time. Once it's become a plasma cell, it's specialized in secreting antibody. But if you get a tetanus shot, you can still detect antibody getting secreted for easily 10 years after. So we know these plasma cells are capable of sticking around for a very long time. But there are non-differentiated B cells as well, called memory B cells, that we also think stick around for a very long time after you have infection to help boost that second infection. Okay, so a closer look at how this happens. So remember, it's the variable region of this immunoglobulin receptor, which is the B cell receptor, that's going to bind to its epitope. It's going to bind to an epitope on the antigen. It's going to engulf it. It's going to process it, load it onto an MHC, and present to the T cell. When this happens, it gets the B cell going. So the secreted antibody, remember at this point, the B cell is going to decide to chop off that tail that attaches it to the membrane, and it's going to secrete the exact same antibody or one that's undergone somatic hypermutation. And the secreted form is therefore also going to be specific for this antigen that's activated it, right? So that's how you know you're activating the right B cell, and it's not an antibody that's going to attack self or anything like that. It's an antibody that's specific for the antigens that are present during a time of inflammation. So that's how this all has to come together. What can happen, though, is that you can have linked recognition. And in this case, you are still going to generate an antibody that is specific for the antigen that has been recognized. But it's not the same antigen that's been presented to the T cell to get it activated. Okay? So what's presented on this MHC is a protein. It's necessarily a protein. It's always a protein. But you don't necessarily want to limit the immune system to only recognizing proteins. So in some cases, you have a protein that's closely associated with, say, a polysaccharide, a non-protein. And it's the non-protein that gets recognized by the antibody. And when this happens, the antibody, again, engulfs, processes, but of course it's a protein that is loaded onto the MHC. The T cell sees it, activates that B cell, and then what the B cell is going to secrete is antibody that is specific for what it recognized in the first place, which is this polysaccharide. Okay, so it's not necessarily the protein. Now this is important for many different things. So there's something called a hapten. A hapten, it would be a non-protein that itself cannot stimulate the immune system, but if it's linked to a protein, then in this linked recognition, it can actually activate and create antibodies specific for it, right? So we exploit this for things like vaccines, right? If we know something that has a capsule, encapsulated bacteria, that could not go through this process alone, we link it to a protein, or we conjugate it to a protein in order to create an immune response, particularly in little kids, because little kids have a very difficult time responding to polysaccharides. So if we link it to proteins, it goes through this system, and it creates an antibody specific to that polysaccharide, but not because of going through a completely T-cell independent way. It's going through this T-cell dependent way because it's exploiting this protein. It is also one of the reasons that a lot of people have reactions to penicillin. Penicillin is the hapten, but it links to a protein in your body and sets off this reaction. Okay. So just like any interaction, don't think of what's going on in isolation. So when a T cell and B cell interact, like I said many times, you have the T cell and T cell receptor, T cell co-receptor with the B cell receptor. You have that CD40, CD40 ligand, but then there's lots of other interactions that have to occur to hold these together to get that synapse. One of the things that helps it adhere is an interaction between LFA1 and ICAM1. So it's an interaction we've talked about before. They also bring up an interaction between CD30 ligand and CD30. But when all this happens, it causes everything to line up. And last week we talked about a immune deficiency that prevents all these from lining up. Remember what it was in the T cell? Wiskott-Aldrich. It's the Wiskott-Aldrich protein that helps these all line up and rearrange. And if you have Wiskott-Aldrich, so it's a T cell problem, but it's not a severe combined immune deficiency because this only happens in T cells that have been activated. That initial response isn't affected. It's the secondary response that's affected, so it's not quite as severe as SCID. I mean, SCIDs are still sick, obviously, but you do have some T cell function. Okay, so here are some pictures of that entire response happening again. So, right, so you have these opsonized antigens that enter the lymph nodes through the lymph, and they can bind to these complement receptors on things like the follicular dendritic cells. The follicular dendritic cells are going to present to the B cell, and it's not going to completely activate them, but it's going to cause some changes. Yeah. Right, it's going to cause some changes. It's going to make them aware that they want to get activated. So they want to travel to the T cell zone. So if a B cell can recognize this antigen, right, it wants to travel to the T cell zone to get fully activated by that T cell. So what has to happen is the address on the B cell has to change to send it somewhere else. So we've already learned what sends things to the T cell zone, right, CCR7. So what has to happen? So what has told it to go to the B cell zone, which is CXCR5, are going to go down on the B cells, and CCR7 are going to go up, so that B cell is going to start traveling towards the T cell zone. Okay? And it's not an on-off switch. It's not that it's going to just suddenly take down all of CXCR5 and put up a bunch of CCR7. It's going to stay somewhere in the middle, so it kind of ends up in this in-between area. It's going to have kind of opposing signals. Right? At the same time, the T cells that have been regulated that have been activated are going to take down some of their CCR7 and put up some CXCR5. So it's also going to start migrating to this kind of in-between zone. So it's going to have these opposing forces that are going to cause it to kind of sit here. This is going to allow the T cell to interact with the B cell. Once that happens, then the B cell can put down its CCR7 and put back up its CXCR5, and then go back to the B cell zone where it can start its follicle. Oh, and I did want to mention, so we talked about this in context of the T cell, but similarly the B cell, once it's been activated, you want it to stay in the lymph node, right? So you don't want it to leave the lymph node now because it needs to proliferate and undergo affinity maturation. So any ideas what receptor gets down-regulated to make it stay in the lymph node? That the CYP receptor. So the CYP receptor is the one that guides it out of the lymph node. So it wants to take away those receptors and it wants to sit in there. So it's just a little bit of up and down to respond to those chemokine signals to stay in the lymph node now. So once it's been activated, it's going to start turning into a plasmablast, where it's going to start secreting antibody. And of course, the first antibody it's secreting is necessarily IgM, and it's not going to be that specific for the antigen it's seen. It's going to make up for that fault because IgM is pentameric. There's going to be 5 hooked together, so it can really, even though it can't grab on very tightly, it's going to have a high avidity. There's going to be lots and lots of different places. It can bind potentially in 10 different times to that antigen, right? Because there's going to be 10, if there's 5 antibodies, there's going to be 10 different variable regions that it can bind to. So it's still going to be fairly effective. But in this stage, it's going to undergo somatic hypermutations to promote this process of affinity maturation, where it then can become a plasma cell. Once it's satisfied that it's really making a good functional antibody that is quite specific, then it can undergo class switching, and it can basically do its job of secreting antibody. So once these B cells have been activated, it goes back to the B cell zone through those addresses, and it starts a germinal center. And what it's going to do is it's going to start dividing, or a few of them are going to seed the primary follicle, and it's going to push other B cells out of the way, and those ones it's pushing out of the way is going to become the mantle zone. And it's going to start proliferating a lot, and when they're going to be very tightly packed together, so it's going to make a dark zone, and then every once in a while, one of these B cells is going to kind of sneak out to get tested, and that's going to be the light zone of these follicles. So here's a picture of this, a drawing of the mantle zone. So these are B cells that are getting pushed away by the proliferating B cells. Here you're going to have your light zone, your dark zone, where they're going to be very dense. It's going to be very rapidly dividing. And then out here in the light zone is where there are going to be follicular dendritic cells that's displaying the antigen. So this is where it's going to get tested. So it's undergoing somatic hypermutations, some of which are going to be not more specific than it was before, so it will no longer recognize this antigen that's been displaying. So it's not going to receive a signal through its receptor, and if it doesn't receive a signal, then it's going to die by apoptosis. If it is more specific, then it's going to come back down, it's going to proliferate, undergo more mutations, it's going to come back out and get tested again. So if it's less specific, it will die because it won't receive a signal. If it's more specific, those will be signaled. A signal will go through the B cell receptor because it will recognize it. It will therefore live and then cycle through this process again and again until it becomes more and more and more and more and more specific. And I think this is exactly what I just said, so I'm not going to perseverate on it. So once you have a specific antigen, right, you want to make different types. Once you have a specific antibody, what do you want? You want different types of antibody, depending on where the infection is. So if you, have an infection that's in your GI tract, well, you probably want to make IgA, which is very good at helping wash away antigens. So you're going to get signals to the T cell, and these are going to be cytokine signals that is going to cause this DNA to once again kind of turn on its mechanisms to rearrange, splice out another region, and then put on an alpha tail to make it an IgA. Similar with IgG, IgE. So these are some of the cytokine signals that actually direct the class switching. So this is what the T cell is going to secrete. So like TGF-beta is going to induce IgA production. IL-4 will induce IgG1 and IgE production. Interferon will induce IgG3 and IgG2. Okay. Oh, definitely. Anything that is on a table. So think of yourself as a very busy researcher / clinician who is on the board, right? And then they tell you, okay, within 3 weeks you have to write 10 questions. So what are you going to do? And you haven't studied immunology forever. You're going to open up Janeway, and you're going to start looking through the tables, and you're going to try and pull out things. You're not going to read the text, right? Well, that's what I would do if I was on the board. I'm not. So anything that is put into a table in either Abbas or Janeway, yes, there you go, right? Because someone's going to flip through, and they're going to say, oh, look at this table. Oh, yes, this is perfect. Perfect. Okay. Right, so this is the system and the process that makes most antibody and the most effective antibody. But there are, like I mentioned before, 2 ways which this can happen without T cells. So it would be a T cell independent response. So one of them is called a TI1 response, or thymic independent type 1 antigen, which would also be known as mitogens. Remember, mitogens are going to force the cell into mitosis despite its specificity. So in the case of like LPS, which would be a B cell mitogen, it's going to bind to toll-like receptors or other pattern recognition receptors on the outside of the cell and stimulate these B cells in a very non-specific way. So theoretically, all kinds of B cells of multiple specificities can get stimulated and can start making antibody. In practice, we don't think this really happens because B cells in general would have a low level of pattern recognition receptors on its surface unless it's already been stimulated in an antigen-specific manner. So a B cell that recognizes a certain antigen will see it and will upregulate all those pattern recognition receptors. So then those B cells are actually favored to divide or get activated to create antibody. So although in theory, right, it's antigen non-specific, it ends up being kind of antigen specific because those are the B cells that are going to be primed and ready to recognize these patterns. Make sense? And then there is a separate way called a TI2 response, a T cell or a thymic independent response. So this happens because multiple B cell receptors will recognize a polyvalent antigen. So if this all didn't exist and there was just one interaction with an antibody, it's going to send a very weak signal to the nucleus, okay? And if it doesn't have a co-receptor, and remember the co-receptor for B cells would be CD19, CD21, co-receptor, one interaction cannot generate a strong enough signal to get the B cell to do anything. But we think that if many, many, many antibody receptors are ligated at the same time, all these weak signals eventually generate a strong enough signal to let it produce a little bit of immunoglobulin. And we think that you can even get a little bit of class switching augmented by this BAS interaction with APRIL, which we've kind of talked about before. This mostly happens in those type of B cells that kind of bridge the gap between innate and adaptive. Mostly makes IgM, these natural antibodies. Definitely not as good as the thymic-dependent manner, but can happen. There is a nice table I'm not going to go over on page 407, figure 1020, that does talk about the different classes of antigens, and it has examples of antigens that can stimulate these different responses. Okay, the last part I thought was pretty straightforward and simple. I'm going to highlight just a couple things. But once class switching has occurred, of course, there's one type of IgM. It is a pentamer. IgD we really don't know that much about. And then IgG, of course, has 4 subclasses, all which will have a little bit different function, some which were great at activating complement, some which are better at opsonizing. IgA just has one. It's secreted as a dimer. And then IgE, like IgG, is a monomer and excels at sensitizing mast cells. Okay, right. So the first function that can happen is neutralization. Well, maybe it talks about opsonization first. I actually don't remember. Regardless. Right, so it can, no, I think it talks about neutralization first. Okay, regardless. So these antibodies can neutralize, and they do this by actually binding to the antigen and preventing it from doing whatever the antigen sort of set out to do in order to infect, right, the human body. Yeah, okay, so I forgot this first. Step back. So, right, IgA is a dimer, and it does have one kind of special things to know about that I would know about. So I said IgM is a pentamer, IgG and IgE are monomers, and IgA is a dimer. So it's a dimer, and it's held together by this J chain. The 2 IgAs are held together by a J chain. But this is made in the mucosa, and it needs to get to the lumen. And this happens through association with something called a poly-Ig receptor, or a polymeric immunoglobulin receptor. That's the PIGR. Everything is capital R, it's a receptor. Right, so it's going to link, and it's going to get endocytosed, transport across the cell, and spit out. So remember, cells actually have to squeeze between 2 cells when they travel through a wall like this. But IgA, it happens in a different way. So it binds to this receptor, and it gets secreted out, and then this gets cleaved off, and the secretory, it becomes, poly-Ig receptor has a secretory component, or it's called a secretory component once it's kind of cleaved off from the membrane. And the secretory component actually has some neutralization abilities on its own. But that's how these IgA dimers, which are still a dimer and still held together by this J chain, then get to the lumen where it can bind to the toxin or the bacteria directly in order to help sweep it out without doing anything infective. There's one other transport receptor that you need to know about, and it's called the neonatal Fc receptor. So this basically does the same thing, but it happens, it helps transfer IgG across the placenta. So a neonatal Fc receptor, 2 of them will bind to one IgG, and it will transport it across the placenta so that it can actually circulate through the baby's circulatory system. And of course, when baby's born, they have all of mom's IgG. They're not making much on their own. But the half-life of IgG is 4 weeks. So right, whenever you dose a medication, you dose it every half-life. So you dose immunoglobulin replacement if you're giving it IV every half-life, so every month you give it. And it's the same when you think about a medication, right? It washes out after 5 to 6 half-lives. The same way IgG from baby, mom's IgG in baby is gone 5 or 6 half-lives, 5 or 6 months. That happens in, just how we think of medication pharmacology. But that doesn't mean that after 5 to 6 months, mom's IgG is going to turn off and baby's is going to turn on. Well, baby starts making a little bit right away, and then they sort of kind of cross over, and then baby's going to have a lot. But you know definitively after 5 or 6 months, if there is IgG there, baby's making it, because mom's would have washed out by then. IgA, of course, is secreted in breast milk. And we think that there are some proteins, like food proteins, that get bound to IgG and are secreted through mom's breast milk at the same time, but maybe not free IgG. Yeah, okay, back to where I wanted to be a minute ago. So depending on the type of infection, the antibody works in very similar ways. So the book talks about toxins, right? So toxins are going to bind to a receptor and then dissociate. So what's going to happen is antibodies are going to come along, and it's going to bind and block the ability of the toxins to get to the receptor in the first place. It has a nice table of lots of different diseases that secrete toxins and what the effect would be. I'm not going to go over those. Does the exact same thing for viruses, right? So for viruses to work, they have to bind to a receptor, exploit a receptor in the outside of a cell, be engulfed, and then it actually uses the DNA machinery in the cell to replicate and make more. So the antibodies are going to neutralize by blocking, by binding to, and not allowing this to bind. So there's a concept called steric hindrance, which I don't think was mentioned in this chapter, but just a word to be comfortable with, which means it's going to bind and it's going to physically block. It's going to hinder the ability to block because there's going to be too much gunk around it for it to be able to come in close proximity enough to actually ligate to that receptor. I think the final example they give would be a bacteria that wants to bind to a receptor in the same way. The antibodies neutralize it. So the next thing it can do is to activate complement. Antibody activates complement, and it does this through the classic pathway of complement activation. The classic pathway is called the classic pathway, not because it's the oldest, it's probably not, it's the first one that we kind of knew about. And the two types of antibodies that can do this are IgM and IgG. It's on that table that we looked at just a minute ago. IgM being the best at fixing complement. The reason being a monomeric antibody on its own is not enough to get this C1q to bind. So it's C1 complex that's going to bind to the antibody. And C1, remember, is made up of C1q, R and S. It's the C1q portion that's going to bind to the antibody. But it has to bind to multiple antibodies. Well, it's a natural fit for IgM because it's a pentamer, so one IgM pentamer can bind to complement and start the cascade. It takes at least 2 IgG, if not more, in order to bind the C1q to get it to stick and start the complement system. Then remember, once the C1q has sat down, what it's going to do, it's going to cleave C2 and C4 to make that C3 convertase. A C3 is going to bind to a C2, C4 complex, and it's going to be a C5 convertase. And that's going to start cleaving to make that MAC complex. So it mentions briefly the ability in solution for antibodies to actually bind to antigen, and depending on the ratio of this happening, it can form these immune complexes, which can bind to complement. And that in and of itself can help clear some of this antigen. It will form these complexes, and it will get essentially opsonized in phagocytosed. We know, though, that in some cases, particularly in autoimmune disease, you can run into trouble with these big complexes doing things like clogging up the kidneys if they're not getting cleared efficiently. Last but not least, antibody opsonizes. So it coats, and then it uses its Fc receptor to bind to a receptor on a phagocyte to help it know to phagocytose that antigen. So the Fc receptor, the Fc portion of the antibody is going to bind to an Fc receptor, and we have lots of choices. And I think we've talked about this nomenclature before. Remember that capital R always stands for receptor. So it's an Fc receptor, meaning it binds to the Fc portion of antibody. And then there's a Greek letter that tells you what type of antibody it binds to. So a simple one is Fc epsilon R1. You know that it binds to the Fc portion of IgE, right? So it's a receptor that binds to the Fc portion of IgE, Fc epsilon R1. There's also a second type of IgE receptor, Fc epsilon R2, CD23. That's what Marcy Chan studies. And then there's, of course, Fc alpha receptors, and then there's Fc gamma receptors. So the gamma receptors march along CD64, 32, and CD16. So Fc gamma R3, Fc gamma R1. But then there's also Fc gamma R2, CD32. So 16, 32, 64. That's how I remember these. They're doubling. The Fc gamma R2 is inhibitory, and you definitely need to know that. Instead of having an ITAM, it has an ITIM, those inhibitory motifs on the inside. Fc gamma R1 and R3 actually has two chains. So it has an alpha chain that is actually associating with the Fc portion, but the signaling part is made of a gamma chain. Fc epsilon R1 receptor is actually very big. It has an alpha, it has a beta subunit that crosses 1, 2, 3, 4 times the membrane, and then a gamma portion that's actually going to do the signaling. Right, so there's a couple different things that can happen here. So the bacterium, the antigen is going to get coated by antibody. It is also likely to get opsonized and coated by complement fragments. There's receptors for both of these on the outside of the cell. Once these receptors engage, as you're seeing here, so this would be an Fc receptor, this would be a complement receptor. It's signaling to undergo phagocytosis, and then of course that phagosome binds with a lysosome, and once that fusion happens, that phagocyte oxidase complex turns on, or NADPH oxidase turns on, and it's, this is not strictly correct, but it's going to turn oxygen into hydrogen peroxide, which of course kills bacteria. We know that because we do that when you have a cut. I think it's more correct to say that the phagocyte oxidase takes oxygen and makes it into reactive oxygen species, which kill this bacteria. So if you can't fuse a phagosome with the lysosome because of mutation list, you have something called Chidia-Higashi syndrome, right? And if you can fuse, but then the NADPH oxidase can't change that oxygen into those reactive oxygen species, you have chronic granulomatous disease. So then, although this bacteria has been engulfed, you can't break it down. You can't officially kill it, just kind of lives there in a stalemate. Okay. Couple last things to mention. So there are some antigens that are going to be too big to get engulfed into a phagosome. An example would be a helminthic parasite. And the thick skin on the helminthic parasite isn't really affected by those reactive oxygen species. It doesn't kind of get chewed through by that phagocyte. So something different has to happen, and primarily the antibody at this point would be IgE. It's going to bind to things like a mast cell or an eosinophil, and then those are going to release these pre-made components like histamine, which is going to bring other mediators. In these other mediators and the histamine is going to be better. It's actually going to directly chew through the skin of the helminth. So instead of engulfing, you're still going to ligate the immunoglobulin receptor. Instead of engulfing, this is too big and it's resistant, so it's just going to directly secrete different components at it. These pre-made components, pre-made. Almost every other part of the immune system has to signal to the nucleus, and the nucleus has to turn on and make protein. It takes a long time, 4 to 6 hours at least. Mast cells are the one thing. Mast cells, they have pre-formed, so it happens very quickly, 15 minutes, an allergic reaction, just bam, it happens. Nothing has to be signaled to the nucleus, nothing new has to be made. It's there, it's ready to go. In the case of a viral infection, one thing that may get turned on would be an NK cell. And we haven't talked, well, I guess we've talked about NK cells a little bit this year. So an NK cell would kind of is almost the innate precursor to cytotoxic T lymphocytes. So they're going to do the same thing. They detect through a series of interactions with the cell whether it's infected or not, or a tumor cell and not a healthy cell, and it's going to send signaling, killing signals to that cell to kill it. So in the case of a viral infection, the NK cell may come in because it's innate. It's already, nothing has to switch. It's already there. It's ready to go, and it kind of does the job until those CD8 positive T cells can come in. So a macrophage kind of gets the job done until other phagocytes like neutrophils have time to come into the site of infection and kill. Mast cells kind of get the job done until eosinophils have time to come in. NK cells kind of get the job done until CD8 T cells come in and have time to do this. But they actually kill the whole cell. The cell's infected, you can't take chances with it anymore, so they kill it by apoptosis. This is called antibody-dependent cell-mediated cytotoxicity, or ADCC. All right, I think that is it. Any questions?
What is the cell actually coated with that, like in that ADCC? So the target cell is a self cell, right, that's infected with the virus?
Correct.
So it's the antibody is recognizing.
So the antibody would be recognizing antigen that is actually on the surface,
okay.
Surface antibody. Surface antigen.
Yeah.
All right.